This is the process of muscular contraction beginning with neuromuscular junction, which is the point at which a nerve fiber contacts a muscle cell. So the neuron and muscle are not actually connected, as you can see here. There's a space called the synaptic cleft in between them, and the action potential reaches the muscle by traveling through this gap using neurotransmitters. And in this case, the neurotransmitters are called acetylcholine, and acetylcholine is what allows the ions to reach the muscle. The receptors on the end of the muscle are called ligand-gated sodium channels, and the channels on the neuron are called calcium-gated channels. The vesicles that are inside the neuron are called synaptic vesicles, and they hold the acetylcholine before it's released, which is the specific neurotransmitter used during neuromuscular junction. The ions on the outside of the muscle want to come in to start the process of contraction, but they are blocked by the channels. Before the action potential reaches the bulb, the interior membrane is more negative than the outside. Once the action potential reaches the bulb, it will change it to be more positive. The change in charge opens the calcium channels on the neuron, allowing calcium ions to come inside the neuron from the outside, also called a calcium influx. So here you see the calcium channels opening up to allow the calcium ions to enter into the neuron. The vesicles release acetylcholine into the synaptic cleft, and once the acetylcholine is released into the synaptic cleft, they bind to the ligand-gated channels on the muscle, allowing them to open. The muscle cell's receiving membrane is called the motor end plate, and when the ligand-gated channels open, sodium ions will pass into the muscle. The sodium influx generates an action potential within the muscle fibers, which essentially causes muscle contraction. So this is a close-up of a muscle fiber. The pink outer layer is called the sarcolemma. The spaghetti noodles are the myofibrils, which we will take a closer look at later. The white piece of styrofoam at the bottom is the nucleus poking out of the bottom, and the Q-tip sticks are the T-tubules. The white tissue paper overlaid the top is the sarcoplasmic reticulum. Each skeletal muscle fiber contains many threads or filaments made primarily of two proteins called actin and myosin. The sarcomere is the band of myosin filaments and actin filaments interlaced to create the subunit of skeletal muscle. So here we are zooming into the myofibrils. I'll have a close-up in just a second. So here's a close-up of the myofibrils. The beads in the middle are the actin, and then the red wax strip is the myosin. The pink pom-poms are the calcium ions that have already been inserted into the muscle, and then so is the yellow pom-pom. And the sarcomere is the band on the top and bottom that interlaces the myosin and actin filaments. So here the myosin heads are attached to the actin filaments, and the myosin tails are attached to the sarcomere. The calcium ions diffuse into the sarcomere and bind to troponin, which are the pink pom-poms. The calcium binding to troponin causes the trypomyosin to move and expose binding sites on actin. The myosin heads latch on firmly to the actin filaments in the overlapping region, forming attachments between the filaments called cross bridges, which are the green things. So now here comes the power stroke. ADP and phosphate are released and cause the myosin heads to move and pull on actin. So the ADP and phosphate and the power stroke are an essential part of muscle contraction, but ATP is also just as important because ATP is the source of energy that causes the myosin heads to release actin. ATP releases energy and reverts to ADP and phosphate. Calcium ions are released from troponin, which causes trypomyosin to cover the binding sites on actin. Muscle contraction continues until the nerve impulse stops and calcium ions are no longer released. So here starts the process of the end of contraction and into relaxation. After contraction, the acetylcholine detaches from the ligand-gated sodium channels and moves back into the synaptic cleft. acetylcholinesterase, the enzyme that breaks down acetylcholine, breaks down ACH into two substances, choline and acetate. After these two are broken down, they're used to create more acetylcholine and the process restarts.